Okay, awesome. Looks like there's a couple people in here right now. There's a chat bar over to the side where you can send me questions. And if for some reason that's not working, you can also email me, or you can send them to the phone number that I have listed in the syllabus. So with that, I did get some original prompts for things that people wanted me to start with reviewing. So let's go ahead and start with those. Okay, so we're looking like we want to do some Punnett squares, maybe some XY. Okay, and sex-linked disorders, things that are recessive, stuff like that. Okay, let me get to, there we go. Awesome, okay, so I'm able to see everybody's questions, so that's fantastic. So I'm actually going to start with transcription translation. So when you're asked to translate or transcribe a DNA sequence, that's going to be whether you're turning it into RNA or whether you're turning it into protein. So if you're given a certain DNA sequence, you're going to have to go from DNA to RNA to protein. So I'm going to screen share some slides here so that you can see what I'm talking about. Let me go ahead and pull those up. All right. Okay, so we got those slides. All right, here we go. So what we're dealing with here, okay, so we've got this information of cells making instructions, okay, so they have their instructions in their DNA, and we're going from DNA to RNA to amino acids, and we have to do this middle step. We can't just jump from DNA to protein, right? The example that we've been using in class is talking about this is a cookbook, right? So if this DNA is a cookbook that your friend has, you can't just take her entire cookbook if you want to take, one recipe. You're going to have to write a little note to yourself and then take that home, and then that you can use to cook, whatever it is, or as Heather said, make cookies. So if you're asked to transcribe a sequence of DNA, you're going to transcribe it from DNA into RNA. And DNA, remember, we're going to read from 3 to 5. And so this AAA, in DNA, A normally pairs with T. But in RNA, we don't have the letter T. In RNA, we only have the letter U. So these 3 A's are going to be paired with UUU. C is still going to pair with G. G is still going to pair with C. And then here we have A pairing with U again. So transcribing is the act of going from DNA to RNA, reading this strand from 3 to 5, making this strand from 5 to 3, seeing that these are U's, these are G's, these are C's, depending on what this top strand is. Translation is going from DNA into RNA and then going from RNA into protein or amino acids. And the way that we do that is that we'll give you this chart right here where we have codons. And these codons are these 3-letter groups, right, where we're always going to start with this AUG. We're always going to start with this AUG, and that's going to code for methionine. And then you're going to look at every 3 letters after that and see what that codes for. So let's look at one of the examples that we had. So here's one of the examples that we did in class, where we had this C, C, T, A, C, G, A, T, etc. as our template strand, and we said, okay, you need to transcribe this, and then you need to translate it. So let me actually go ahead and type in here with what this would be. So our mRNA, if our DNA is running from 3 to 5, our mRNA is going to run from 5 to 3. And our letters, I'm actually going to erase this. Our letters are going to be in a complementary fashion to the DNA, but remember we don't have T in RNA, we only have U. So the complement to C is G, and then another G, and then the complement to T is A, the complement to A is U, the U is currently being covered up because this picture is too big, so the complement to A is U, as we can see right here, the complement to C is G, the complement to G is C, to A is U, T is A, U, U, C, G, U, oh, nope, that's A, U, okay, there we go. This is our mRNA, and it goes like this, and it goes from 5' to 3'. So we're going from 5' to 3', and we're going to look for our start codon. Anytime you have to translate, okay, anytime you have to translate, you're going to look for this start codon, this AUG, and that is super crucial. Because if you start reading right here and you think this is a codon, your sequence is going to be wrong. You cannot start translating until you find this AUG. Okay? This is always going to be your start codon, and we're always going to put it somewhere close to the start of the mRNA. So our AUG is going to be our start codon. Okay, so I'm going to space that out so we know this is one of our codons right here. And then we know every 3 letters after that is also going to be a codon. So we have CUA, and then we have UUC, and then we have GAU, and then we turn those into amino acids. Okay, so for amino acids, AUG, as we can see on this little table right here, AUG is for methionine. CUA over here, there it is, CUA codes for leucine. UUC is up here, and it codes for phenylalanine or Phe. And then GAU codes for asparagine ASP. And that's how you do the translation. If you are asked to translate DNA, you have to go through this intermediate step. If you're asked to translate DNA, you have to transcribe and then translate it. Even if we don't blatantly say transcribe it, you have to transcribe it because you can't just go from DNA to protein, right? You have to go from DNA to RNA into amino acids. If you're asked to transcribe it, the only step you have to do is you have to go from DNA to RNA. Okay, so that's kind of our transcription translation overview. Let me do some quick troubleshooting to make sure that everything is looking okay. Awesome. So it looks pretty good. And okay, so we had, so we did transcription translation. And okay, here's another quick one. Can you go over, oh, hold on. There we go. Okay. Can you go over 5' to 3'? Yes, I can, because that's still in the slides that I'm on. So I'm going to do 5' 3' really quick. Let me find that. All right. So I'm going to go over our basic nucleotide structure again. So when we're talking about 5' and 3', that's just giving an idea of what side of the nucleotide we're dealing with here. Okay. So with our sugar phosphate backbone of DNA, and to back up even further, a reminder that our DNA is made up of, we have these bases in the center that are hydrogen bonding with each other. Then each of these bases is bound to a sugar, and each of these sugars is bound to a phosphate. And so the backbone is we have this phosphate group here that's going to be covalently bonded to a sugar, and then a phosphate that's bound to a sugar, and then a phosphate that's bound to a sugar. And I can actually make this a lot bigger. There we go. Okay, so we've got a phosphate on a sugar, and we've got a phosphate on a sugar. And then off of the sugar is where we have our base. So let's just look at our backbone, because we have some notation here where we say, okay, this is our 5' end. Our 5' end has a phosphate group, and our 3' end has an OH, what we call a hydroxyl group. And this shows that this side is running from 5 to 3, and this other side is running from 3 to 5. So we can actually see this side is flipped upside down in comparison to this side. Right? So for this sugar, the oxygen is up, and for this one, the oxygen is down. Right? And so this side, the 3' end, is up, and the 3' end here is down. But in the nucleus, it's going to just be wound up. It's going to be all, strung around. It's not necessarily going to have an up or down. The way that it's read in the nucleus, how they know to read from one direction or another, has to do with the 3 and the 5' ends. And where that comes from is this numbering of our carbons. So at each of these points, we have a carbon in our notation. We're saying there's a carbon here, there's a carbon here, 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 and here. The carbon that is the closest to the base, we call the first prime carbon. The one that's the second closest to the base is the 2', then the 3', the 4', and the 5'. So when we're talking about our orientation, what we're really talking about is which carbon is up over here or potentially down over here. So when we have all of these sugar rings and phosphate groups all linked together, this 5' carbon here is attached to a phosphate, and this 3' carbon is attached to an OH group. So you can always remember when you're trying to figure out what end is 5' and what end is 3', the 5' end has the phosphate, the 3' end has the hydroxyl. And we give this context for reading in certain directions because if we have these bases over here, we want to know, do you read them in the order of 1, 2, 3, or do you read them 3, 2, 1? And because that's going to give you very different information if you're trying to encode that gene, right? And we know we can only read DNA 3 to 5. Okay? That's the direction that we read it. So if we were trying to read this little bit of DNA right here, we would read it 3, 2, 1. Okay? Directionality in DNA is very similar to the fact that when we read anything that's written, we read from left to right. Right? We read from left to right. If you started reading your textbook right to left, all of the words would be very different. It would make almost no sense. And so that's the same as reading DNA backwards. But in this case, instead of left to right, we're doing 3' to 5'. And so that's our 3 to 5 of our backbone. And, okay. Let's see what else we've got here. So that's our 3', 5'. Awesome. Okay. Okay. I'm getting some information from people. I'm trying to monitor. I have a couple different windows open. I'm trying to monitor the chat. Oh no. And it looks like we're having some more technical issues. Okay, so now that I'm back, I can see the chat. So when I was trying to screen share and I was on the slides, could any of you see anything? Okay, so you couldn't see when I was going over prime. Let me try to screen share again. Thank you for helping me screenshot this, troubleshoot this. This is the first time I've tried going live. Every other time I've done this, I've been doing Google Hangouts, so it's easier to get direct feedback. Okay. Okay, so yeah, it sounds like you can't see it when it's full screen. So I'm going to try to screen share again and go over 3', 5', and we'll see if that works. So I'm going to pull up the window again. Okay, I'm going to share it, and let me know if you can see it now. Awesome. Oh, thank you all for the feedback. All right. So it looks like we can see this. Let's make this a little bit bigger. Okay. All right. So you all just heard what I was talking about, but you had no visuals. So let me go over this again really quick. Okay, so with DNA, we've got our bases in the center that are hydrogen bonding with each other. Then we have our sugar, and then we have our phosphate. So here's our sugar phosphate backbone. And when we talk about our sugar phosphate backbone, we talk about the 5' end versus the 3' end. And here's why that matters. Because if we read this strand of DNA over here from 3 to 5, we're going to read it A, C, G, T in that specific order. If we read it the other way, we would read it T, G, C, A. And those are very different pieces of information, right? Those letters are in completely different direction. So knowing what direction to read it and understanding that DNA has directionality is very important for genes and how we read genes and understand genes and can transcribe and translate those genes. Awesome. All right. So here we have a close-up of our nucleotide. And our nucleotide, our sugar, is made up of 5 carbons, and we number those carbons depending on how close to our nitrogenous base they are. So here we have our first carbon, we call our 1' carbon. And then as we get farther away, we number them 1', 2', 3', 4', 5'. Okay. And so this is the first carbon that's close to the base, second carbon, third carbon, fourth carbon, fifth carbon. That's where these numbers come from, is this one is the third closest carbon to the base, this one is the fifth closest carbon to the base, so this one is number 3', this one is number 5'. And our 5' carbon has the phosphate on it. Our 3' carbon has this OH group on it. And that's how we are going to link these sugars and phosphates together, is we're going to have this O coming off of our 3', and that's going to link to the phosphate group of the next 5', and then here at the 3' we have O coming off, and then our phosphate group linking to our 5', which gives us our reading frame so we know what direction to go in. Okay? So for this example, we need to know if we're going to read these bases in the order of 1, 2, 3, or 3, 2, 1. And because we read DNA from 3 to 5, we're always going to read DNA 3 to 5. We would read these bases in the order 3, 2, 1. So this is where I was talking about like us reading left to right versus right to left. For DNA, instead of left to right, we're reading 3 to 5. Our 3' group is also sometimes going to be where we have our OH. Okay? Sometimes it's just an oxygen, but oftentimes it's an OH group. And our 5' is where we have our phosphate. Okay, so if we're thinking about where our phosphate is, where our 5' end is, the 5' end is going to be where our phosphate is. And the 3' end is where the O is. Awesome. All right. All right, so that's it for 3', 5' structure. Now we're going to switch a little bit and go over, then I'm going to go way back and we'll do genetics. So first, while I'm still in these slides, let's talk about mutations really quick. Whew. All right, so we started here and we started saying that mutations are changes that we have that are going to be different than the normal code. Okay, so the normal nucleotide sequences will encode for genes just fine and they'll have information in them. But if we mess something up, okay, if there's some error in replication or cell division, where right here we have this base changed from what it's supposed to be to something else, or here we lost part of a chromosome, or here we got an extra chromosome, right? And mutations happen all the time. Mutations can happen when you're exposed to X-rays. Mutations can happen when you're exposed to carcinogens like tobacco smoke. They can happen when you're exposed to UV radiation. Right? Mutations can happen in your DNA, and a lot of the time they don't have any effects, but sometimes they do. And so here we're going to talk about the different types of mutations and kind of these ideas of what different types of mutations we have, when they're going to have effects, and what those effects are. So here we have point mutations, and mostly what we're going to be talking about is we're going to be talking about point mutations, right? Back on this slide, we said you can lose an entire chunk of a chromosome. we're not really going to address what happens if you lose a whole leg of a chromosome. We're going to focus in on more of this. We're changing 1 to 3 or maybe 5 bases. So it's just this one single base here is changing from a T to an A. And when we change from a T to an A, this mRNA is also going to change, and this hemoglobin amino acid is also going to change. Okay, so this point mutation is just this one base changes, and we're actually changing the protein that we make because we change this one base. So for our point mutations, we have a substitution type mutation where we're substituting something for something else. Right, so, oh dear, I lost my screen. There we go. So here, okay, for our substitution, this normally is supposed to be a G, and if it changes to an A, that's going to change this amino acid from a glycine to a serine. So this amino acid is actually going to change because this nucleotide is now different. We can also have deletions and insertions. And like we talked about today, deletions and insertions are going to be a lot of times have a much bigger impact on the sequence because we can shift our entire reading frame. Okay, so let's look at both of these a little bit closer. So for a substitution mutation, we can have a silent, a nonsense, or a missense mutation. So our normal sequence is that we have in our DNA, we have TTC, which makes our mRNA AAG, which makes the amino acid we're coding for lysine. A silent mutation is when one of these bases changes. So it's different from the non-mutated version, but overall it doesn't change anything. Okay, because of the fact that amino acids are coded for by multiple codons. So here we can actually see that AAG right here, okay, codes for lysine, and it's also coded for by AAA. Okay, so if your codon is AAA or AAG, it's going to code for lysine. No problem. And here we see we change from AAG to AAG to AAA, which makes our lysine still lysine. Technically, there's a mutation, but we call it a silent mutation because it doesn't show, right? You would not know that you had a mutation in this protein because the protein is going to look exactly the same. A nonsense mutation is if we have this TTC change into ATC, which makes this mRNA UAG, and that is a stop signal. Okay, so this signals the ribosome to absolutely stop translating the mRNA. Normally, we're supposed to keep going and keep putting amino acids on for however long, but if you get this stop codon too early, it's going to stop making that protein, and you're going to have a pretty much non-functional protein. Okay, nonsense means it's a extension of no sense, in that there is no more amino acids coming after this. Okay, a nonsense mutation is always when the codon mutates in order to be a stop codon. Missense is when we change the amino acid. So a missense is this mutates, okay, this original sequence mutates, and then it's changing from TTC to TCC or TGC or some other codon where now instead of lysine, we have arginine. Or now instead of lysine, we have, I think this is threonine. I'm not as good in my amino acids as Heather is. Okay. But instead of having lysine here, we end up having a different amino acid. That's our missense. And these are all substitution mutations. We're changing this C, okay, and turning it into a T, or we're changing this T and turning it into an A, or this T into a C, or something like that. Okay, where we're just taking one letter and we're changing it. Insertion or deletion, we're either putting in a new letter or taking out a letter. And there are two types of mutations for insertion deletion. We have frame shift and non-frame shift. So a frame shift mutation for an insertion would be we put in one letter, and then now that bumps this codon, which is supposed to be CCC, bumps it over. So it's actually got two of the C's right here, and the next C is in the next codon. Okay? So now instead of having CCC, we have TCC and then CAC, and then this T is over here by itself, which changes our amino acid sequence from GLN pro THR to GLN SER HIS, which is a very different sequence than we started with. Non-frameshift mutations happen if you insert or delete letters 3 at a time. So if we insert 3 brand new letters, we're still going to preserve our 3-letter reading frame. Right? We're going to shove this entire brand new codon in here, so that's still a mutation. We're changing the original sequence. But our overall GLN, PRO, THR, it's still there. It's just got another amino acid in there. So non-frameshift is when our codons are still preserved. There's just been 1 or 2 codons inserted or removed from that original sequence. A frameshift is we're putting in 1 or 2 letters and it's bumping everything over. Okay, couple quick addressments. I'm awake, it's fine. Couple quick addressing some of the questions in the text chat. So for the information that corresponds DNA to mRNA, I will not give you that information. You will not have it. You need to know that C and G bond. You need to know that A and U bond. You also need to know that if you're doing DNA replication, it's A and T will bond together, and C and G will bond together. Okay, so A and T will bond. A will either bond with T if you're making DNA, or it'll bond with U if you're making RNA. Okay. You do not know if you do not have to study chapter 11. Okay? Chapter 11 will not be on the exam. We had, I think, 2 slides on it today, and it's basically the take-home message. Your genes can be turned on or off. Right? Not all your genes are on all the time. The big take-home from 11 is that they can be on or off. All right. Let's go back up here. So there's a lot of questions coming in for classical genetics and things like that. Okay. So let me go ahead and switch gears a little bit and we'll go over there. Let's do, how do I want to do this? Let's do some Punnett squares. Okay. Also, the more specific that you are with questions, the easier it is for me to answer them. Requests like, can you go over meiosis? Meiosis, we took a long time to talk about in lab, and so that's kind of tough to go over the whole thing. If there's a specific part of it that you're confused about, a specific slide number, or a specific term, that really helps me in answering questions. Okay, so give me 1 second to go over some classical genetics. I'm just setting up a prompt right now, so I might go quiet for a second. Awesome. Okay, so let's switch screen sharing. Oh, look, it's my face. Okay, we're going to screen share, and we're going to screen share a different screen. We're going to screen share this one. All right. Okay, so we've got a pure breeding black cat and a pure breeding orange cat are bred together, and all their offspring are orange. We have to figure out what trait is dominant, and we have to figure out a Punnett square for this cross. So when we say pure breeding here, okay, this term, pure breeding, is telling us something. If something is pure breeding, that means it's homozygous. Okay, so we've got two instances of pure breeding here where we're dealing with, okay, we know that these are homozygous because they can only have one type of the allele in order to be pure breeding, right? If you cross a heterozygote to a heterozygote, you're going to get a variety of phenotypes in the offspring. If something is pure breeding, it is homozygous. It only has one option for what allele it can give. So let's get this first question out of the way, and then we'll go through all the steps. The 5 steps that your lab manual takes you through are going to be really handy for solving genetics problems. So a pure breeding black cat and a pure breeding orange cat. So this one only has the allele for black fur. This one only has the allele for orange fur. All their offspring are orange. So we're crossing one homozygous cat with another homozygous cat, which means all their offspring are going to be heterozygous, and we'll prove that with a Punnett square in a second. But if all their offspring are orange, that means that only one trait is showing, right? They're not black and orange. They're not a mix. They're just orange, which means the dominant trait is orange. Let's go on to our Punnett square for this cross. In order to do a Punnett square, you can't just jump into the Punnett square. You've got to do some setup first. So the first thing we're going to do is we're going to choose our letters. And so I'm going to do F for fur. So I'm going to do big F, which is dominant, and we know that orange is dominant. Okay, big F is going to be an orange. Little f is going to mean black. The next thing is to figure out the genotypes of our parents using the letters from step 1. So let's see, pure breeding black cat. Let's go ahead and make this one mom. We'll make this one dad. So mom is a pure breeding black cat. Pure breeding meaning homozygous. And we said, because of the notation we used up here, the letters that we're using is we're using big F for orange and little f for black. So mom's genotype is going to be little f, little f. She has to have 2 letters because she's diploid. Okay, so cats are diploid. She's going to have both copies as little f. Dad is going to be big F, because he's homozygous for orange. Then we have to, let's see, determine the gametes of each parent. Move our Punnett square down a little bit. Okay, determine the gametes of each parent. Mom can give only one option, right? Mom can give little f, or mom can give little f. That's it. Mom can give a little f. Dad can give big F or big F. Dad can only give big. Okay. So that's it. Those are our gametes. The next step is to set up the Punnett square. And when we set up the Punnett square, we're going to put dad on this side, and we're going to put mom on this side. Okay, so dad's gametes go in this part of the table, and mom's go over here. Okay, so I'm going to put this notation. Oh, no autocorrect. It's very important that you don't do that. Okay. So these little f's come from mom, and these big ones come from dad. Okay. Then we fill in our Punnett square, which is down here, to figure out, okay, you can either get this big F from dad, or this with this little f from mom, or if you get this big F from dad, you can get this little f from mom. If you get this one, you can get it with that one. Or if you get this one, you can also get it with this one. It doesn't matter, they're all the same. Okay. Everybody in this F1 generation is going to be a heterozygote. And that's our last step for this. Fill in the Punnett square. Okay, so those are our 5 steps. So we did those. Okay, so here's our F1 generation. F is in filial, the first offspring generation. And now that we have our F1 generation, let's figure out our F2 generation. So our F2 generation, we make by crossing an F1 to another F1. So what will the genotypic and phenotypic ratios of the F2 generation be? We're going to do the same 5 steps that we did before. Okay? So first step is going to be determine our letters, which we already did. We said big F is for orange, little f is for black. And then we said we had to get our numbering system correct. There we go. That's not right. None of this is right. Oh dear. Love troubleshooting while we're live. All right, so we've got our letters, and then we have to determine the genotypes of the parents. Now, when we're doing an F2 generation, the genotypes of the parents are going to be the offspring from the F1 generation. So we already have our genotypes. This is our genotype right here. So the genotype of both mom and dad is going to be a heterozygote. Okay, so mom is going to be big, little, dad is going to be big, little. Now we have to figure out to determine the gametes of each parent. Okay. So the gametes of each parent, let's do mom first. So mom can either give this big F, or the other option is she could give that little f. Dad could give his big F, or he could give his little f. So those are our gametes. And now we set up our Punnett square. I'm just going to steal this one. So now we set up our Punnett square with our gametes, either across the top or the side. And someone asked, how many squares do you use to set up a Punnett square? Unless you're doing a cross with more than one gene, you're always going to have 4 squares that you'll fill in for your Punnett square. For ease of filling it in, I have more squares over here so I can show you very clearly what I'm correlating with these inner squares. Okay, but this is just for my purposes in this document. These 4 squares right here are the ones that really matter. Okay, these are the 4 squares that you're going to be drawing if you're drawing out a Punnett square on the exam. This is for one gene. Okay, so we're going to put dad's gametes in the top. Okay, so dad can give big F, as we said, or he can give the little. Mom can also give big, okay, or she can give little. Then we can fill this in and we see, okay, you can get a big F from dad and a big F from mom, or you can get a big F from mom and a little f from dad, or a big F from dad and a little f from mom, or a little from dad and a little from mom. So these are all of our options, and that's our last step, right, is fill in the Punnett square. So we fill in our Punnett square, looks fantastic. And now we have to go back up to our original question. Okay, what are our genotypic and phenotypic ratios? So our ratios are dealing with how many of this do we have compared to how many of this compared to how many of that. Okay, so our genotypic ratio is going to be dealing with the genotypes. What genotypes do we have and how many of them are there? Our genotypic ratio is that we have one of these. So one double dominant, one homozygous dominant, and then we draw a colon. Okay. We have one big F, and then we have 2 heterozygotes. We have this heterozygote and we have this heterozygote. So 2 big little, and we have 1 little f, little f. Okay, so we have 1 homozygous recessive. That's our genotypic ratio. The genotypic ratio is always looking at what are the genotypes and how many of them are there compared to the other types. The phenotypic ratio has to do with what do these offspring actually look like? Okay, what are our offspring actually going to look like? And we can go back up to know right here, okay, the dominant trait is orange and the recessive trait is black. So this baby, okay, this little kitten is going to be orange. Okay, so this one is going to be orange. That's not orange. That's orange. Okay, this one is going to be orange. This one also has one of those dominant alleles, so that one's going to be orange. This one has one of the dominant alleles, so that's going to be orange. And this one has both recessive, which means it's going to be black. Phenotypic ratios, as in what are the phenotypes of these individuals? What do they look like? And we have 1, 2, 3 that are orange, to 1 that is black. Okay. And these ratios are the simplified version of these ratios. If these cats had 8 kittens, you would expect to see 6 orange and 2 black. Okay, so if you had a bunch of kittens in a box and you picked up 1 kitten and it was black, you would expect to find 3 other orange kittens for every black kitten, is what we're saying with these ratios. Okay, for every 1 that is homozygous dominant, we expect to find 2 that are heterozygous and 1 that is homozygous recessive. Okay, so that's how we go through our Punnett square. Okay. Oh, okay. So good question. Can I give an example of the product law and the additive law? Okay. So let me switch screens again. And here's my face. All right. Got about 10 minutes left. I'll get through as much as I can. All right. So we're going to switch. We're going to screen share something else. And I'm not going to make it full screen so that you guys can actually see what I'm talking about. Learning has occurred. Okay. So we're going to screen share this one. All right. So we're going back into chapter 9 to do the additive and the product law. Here. All right. So we have 2 different rules when thinking about probability. We have the rule of multiplication and we have the rule of addition. Okay. So our rule of multiplication is any situation where we have the word and being thrown in. Okay. So here we have what's the probability of having 3 boys in a row, where the first child you have to have is a boy, and the second child you have to have is a boy, and the third child you have to have is a boy. That means you're going to multiply the probabilities together. So our example here is with having 3 boys in a row. The probability of having 1 boy at any time is 1/2, right? It's 50-50 if you have a boy or a girl. So we have 1/2 of the first instance of having a boy times 1/2, which is the probability of the second time you have a boy, versus the third time, which is also 1/2, which gives you a 1 in 8 possibility of having 3 boys in a row, right? Because this and this and this has to happen in order for this to be true. The other option is the rule of addition. Okay, so the probability of having a girl and a boy, but not in any particular order. So we can figure out all possible combinations where we can have a girl and a girl, we can have a girl then a boy then a girl, or a boy and a boy. Each of these is going to have a 1/2 probability. And to figure out what's the probability of having a girl and a boy, it's going to be multiplication, because it's girl and then boy, or boy and then girl. Okay? So 1/2 by 1/2. Odds of this happening are 1 in 4. Odds of this happening are 1 in 4, and either option is good for us. Okay? Either option is fine, and so it gives us a 1/2 odds that we'll have a girl than a boy. And we can think about it like this. If you've ever had a friend, who's going on a date, and you kind of chat with them before they leave, and they're all, nervous and excited to meet up with their date, and you say, okay, what do you think is gonna what would make a successful date in your head? And maybe you have one friend who's pretty mellow, and they say, well, if my date brings me flowers, or if they pay for dinner, or if they drive me home, I would consider that a successful date. And the odds of having one of those things happen is pretty high, right? It's just one of those three things could happen, and they would consider it a success, which is the outcome that they're looking for. Versus you have a friend that's, a little bit more honed in on the dating game, and they say, I will only consider the date a success if they bring me flowers and they pay for dinner and they drive me home. The odds of all three of those things happening, it's not going to be as likely that all three will happen. It's more likely that just one of those things will happen, right? And that's where we see with our rule of addition, right? Our odds are higher, right? Of like, oh, a boy and a girl in no particular order, right? This could happen or this could happen, we would call it a success. Those odds are pretty high. Versus if we're saying, no, it has to be a girl and then a boy, no other option, those odds are going to be lower. Okay, so if we have to deal with multiple events happening in a row, right, or multiple things have to be true in order for us to consider it a success or the outcome that we want, we're going to multiply those odds together, and they're going to be smaller than if we have multiple options, right? This or this is fine. Those odds are going to be a little bit higher. Do, do, do. Okay, some quick questions that I can get to. Looks like I got 2 examples of genotypic and phenotypic ratios, which is awesome. I did not know that was a question, and I kind of just slipped that in there. There was a question about transcribing anticodons. You cannot transcribe anticodons. Okay, and here's what I mean by that. Let me change what I'm screen sharing here. Okay. So you cannot transcribe anticodons, and here's what I mean by that. Let me go over to my tRNA. You can translate an mRNA. In order to translate an mRNA, you have to use tRNAs, these transfer RNAs, that have an anticodon portion of them. So what information we could get from you, or we could ask, is we say, okay, if a codon is AUG, what would the anticodon be? Or if the anticodon is UAC, what would the codon be? Or what are 2 of the important parts of a tRNA? It's the anticodon and the amino acid attachment site. Okay, so we're not going to ask you to necessarily transcribe this because you can't transcribe it, and that term really matters. Okay, you can transcribe DNA, which means you're turning it into RNA. Transcription refers to DNA going into RNA. Translation refers to mRNA going into amino acids. tRNAs and anticodons are involved in translation, but what you are actually translating is the mRNA. Okay. Looks like we've got some questions on pedigree. Oh, Sergio, yeah, I'm going to do pedigree right now. And that's not exactly what I meant by F1 and F2. F1 and F2 can be related to pedigree, but it's different. F1 and F2 are what we're predicting. So the F1 and the F2 generations that we did in the genetics problem are kind of what do we expect to see? What are the probabilities that we'll have offspring that look this way or have this genotype? A pedigree is actually who came out as a baby, what did they look like, what did their parents look like, what did their grandparents look like. So let me go ahead and pull up those slides and get a little bit more into detail with pedigrees. Okay, so we're going to jump back into 9. Over here, we left off on multiplication and addition. All right. So the pedigree example that we have over here is we have a couple right here in the middle, okay, that had children. Okay, this horizontal line shows that they mated. Okay, because we can use these also for plants, we can use them for animals, we can use them for humans. Okay, so this horizontal line just indicates that mating has occurred. They have had offspring. The vertical line connects them to their offspring, and this shows that they had 2 girls. Okay, and these girls, because they're connected by lines that go like this, are sisters. Okay. We generally show circles for females, okay, and we use squares for males. The other important notation that we have is we have squares or, sorry, not squares and circles. We have a colored in or closed versus not colored in or open. And that has to do with, are they affected or not? And it depends on what kind of trait we're dealing with. So with this example pedigree, we're dealing with a recessive type trait. Okay? So free earlobes. Okay? So free earlobes, where we have the shaded in being affected. Okay? So shaded in has a free earlobe. Okay? This is a free earlobe, free, free. Open shows not affected, which means in this case, not a free earlobe. We can also use this for other things like Huntington's disease, like blonde hair, like hemophilia. Okay? All kinds of things that we can show through pedigrees. So let's look at what this one is showing. This one is showing we have grandparents on this side, and we have grandparents on this side. So we have this male mating with this female and having 4 offspring. They have a girl, a boy, a boy, and a boy. The offspring are usually shown in order from firstborn to most recent born. So we've got grandpa, grandma have 4 kids. This female is unaffected. These 2 males are affected. This male is unaffected. Other set of grandparents, we have grandpa who is affected, mates with grandma who is not. They have 2 daughters, one who is affected and one who is not. This man and this woman have offspring, and they have 2 daughters. This one is affected and this one is not. The other notation that we've used for this is we've used a half shading over here when we were talking about sex-linked disorders. We used a half shading to show this person is a carrier. Okay? This person has 1 copy of a recessive allele. So they don't show that they have it, but they're carrying it, right? And they're passing it down to their offspring. And in this case, it's X-linked. So if a girl gets 1 copy, she will not show the phenotype. Okay? For X-linked recessive disorders, if a girl gets 1 copy, she is not going to show the phenotype. A girl has to get 2 copies to show the phenotype for either colorblindness or hemophilia is the other common one that we talk about. A male only has to get 1 bad copy of an X to show the phenotype because he does not have a second X. He has 1 X and then he has a Y. We can use pedigrees. Okay? We can take text and convert it into pedigrees. We can then use pedigrees to figure out the genotypes. If you are going to figure out the genotypes by using a pedigree, go for the recessives first, okay? This individual is showing that they are affected, and it's affected for a recessive trait, which means both copies of the alleles they have are both recessive, meaning they had to get one recessive from mom and one recessive from dad. These are not shaded in, which shows you they are not affected, which means their other allele has to be this capital F. They both have to be heterozygotes. You can then trace it back further using their siblings to figure out what all these genotypes are. A really great practice that I would recommend is going and finding some example pedigrees. There should be some on your online resources through your book site. But also you can take this pedigree itself and just delete all of these captions. See if you can figure out what all these genotypes are. Okay? Being able to translate some text about a family history into a pedigree and then translate that pedigree into genotypes is very important. I would recommend that if you don't know how to do that, that you practice through that at least 2 to 3 times tonight, okay? Because that is going to be important. Okay. Okay, for sex-linked disorders, yes, the female is always the carrier. Okay, the female is always going to be the carrier. Men cannot be carriers for a sex-linked disease because they only have one copy of the X. Okay, women can be carriers or they can have it, right? If they get a bad X from mom and they get a bad X from dad, it is possible for them to have that sex-linked trait. Okay, so this one does not show it. That's unfortunate. It's not likely, right? In order for this daughter, Alexandra, to have the sex-linked disorder of hemophilia, she would have to get a bad X from her mother, Alice, and her father, Louis, would have had to have actually had hemophilia. The odds of a male with hemophilia and a female who is a carrier actually having a child together, first of all, finding each other, having a kid, not very likely, as well as they would have to have a daughter who then gets the bad X from both parents, and there's only a 50-50 chance that that will happen. Odds are girls are not going to show X-linked recessive traits. They are more likely to be carriers for X-linked recessive traits than they are to show the disease. It's possible, but it's not very likely. Okay, so there's a quick question on the study guide, asks for 5 of Mendel's theories, and you guys can only find 4. So let me go back up here. Okay. One of his important conclusions, his hypotheses, one of them was that there is genetic variation. Okay, so that's the first one. The next one is that each person will inherit 2 copies of an allele. If you have 2 copies, one of them will be dominant over the other one. That during meiosis, the heritable characters will segregate. Okay, so 2 alleles will segregate from each other. As well as, oh, what is the other one? Segregation and it's independent assortment and crossing over. Let me just double check this really quick. Hold on. We got a very similar question earlier. And if I was less brain dead, I would remember. Okay. Oh, okay. So law of segregation is the law of independent assortment. Let me go ahead and just make another Word document to separate these out for you guys so it's more clear. Okay, give me 1 second to type this up. Here we go. So here's a quick summary of all of Mendel's hypotheses. So one thing that he determined is there is variation. Okay? There's variation in allele options. Okay, so in versions of a gene, there is variation. There's genetic variation that already exists, right? Not everything looks the same. He was looking at pea plants and he saw that you could either be purple or white flowered, you could have green or yellow seeds, you could have inflated or wrinkled pods, like all kinds of variation that he saw. He also figured out that each individual will have 2 copies of a gene. Okay? They will have 2 alleles present. They will have 2 alleles per individual. If they have 2 alleles, if they have 2 different versions, one of them is going to be dominant over the other. Because Mendel only saw complete dominance. Okay? He saw complete dominance, and that one allele is always going to be dominant over another allele. We talked a little bit more about how we've seen some other examples of dominance. We've seen incomplete dominance and we've seen codominance and things like that. He only saw complete dominance. So this is one of his hypotheses. The law of segregation kind of breaks down into 2 different ideas. The law of segregation is that a sperm or an egg cell will only carry 1 allele. Okay? Each parent will have 2 copies, but they only get to give 1 to their offspring. So that's one idea. The other one is that each pair of alleles segregates independently. The inheritance of one trait does not have an effect on the inheritance of a different trait. Okay? So all 5 of these are the 5 hypotheses. The law of segregation that we have in the slides kind of separates out in these 2 different ideas, which is that a parent only gives 1 copy, and that whatever alleles you're inheriting, you inherit them, they get inherited independently of each other. Okay, last quick answer for meiosis. For meiosis, there are 2 really important things that happen in meiosis. Okay, so for mitosis, mitosis is the process of your cells just splitting apart, okay, and making exact copies of each other. So if you want to make an exact copy of a cell, the cell is going to do mitosis, and it's going to go from 1 cell into 2 cells. It's an exact copy. So if the parent cell is diploid, all the offspring cells are diploid. If the parent cell started with 28 chromosomes, both daughter cells are going to have 28 chromosomes. That's going to happen in your hair follicles, that's going to happen on your skin, that's going to happen in the lining of your stomach. Mitosis is going to happen in pretty much all places in your body, except for your reproductive organs. Meiosis happens where gametes are produced. So if gametes are being produced, they're being produced either in the ovaries or in the testes. Meiosis is the process of making gametes, and it's a very specific process where we're going from a diploid cell, and we want to make haploid daughter cells. We're making 4 daughter cells be haploid. The 2 important things that happen in meiosis, because meiosis is 2 rounds of division, right? It would turn 1 cell into 2 and those 2 into 4. There are 2 rounds of division. During the first round of division, there are 2 very important things that happen. One thing is crossing over. Crossing over is when non-homologous, non-sister chromatids of a homologous pair, okay, I'll say that again correctly, non-sister chromatids of a homologous pair form tetrads and exchange genetic information. That's crossing over. And that happens in prophase 1. The other thing that is important is independent assortment. Independent assortment is when the way the tetrads are aligned don't depend on how the other tetrads are aligned. That happens in metaphase 1. So prophase 1 is where we have crossing over. Metaphase 1 is where we have independent assortment. And those 2 things only matter if we're talking about meiosis, not mitosis. Okay? Mitosis is just a very straight Take one thing and you make two things and they're exactly the same. Meiosis is we're making gametes and we want to make them very diverse. We want to have a lot of variation in the gametes, which is why we have crossing over and independent assortment. Okay, I think I got to everything. I'm going to hang out for about 3 more minutes. If there's any other last minute questions, please make sure that you submit them now. And if there are no more questions, then I'm going to log off, and I hope all of you have a wonderful time studying. I'll be up for a little while, so if you want to send me emails with questions, I'll be up for a little while longer. Oh, good last minute question. Is it correct to say that if something has 48 chromosomes, the daughter cells will have 24 after meiosis? Yes. If something starts with 48 chromosomes, after meiosis, the daughter cells will have 24 chromosomes each. All right, looks like we're all done. So for me and my tiny helpers, we're going to say thanks and good night. I hope you all have a wonderful time studying.